Angiosperms. Unlike the gymnosperms, where the ovules are naked in the angiosperms of flowering plants, the pollen grains and ovules are developed in the specialized structure called flowers. In angiosperms, the seeds are enclosed by fruits. The angiosperms are an exceptionally large group of plants occurring in a wide range of habitats. They range in size from tiny, almost microscopic wolfia to tall trees of eucalyptus, over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes, the dicotyledons and the monocotyledons. The dicotyledons are characterized by having two cotyledons in their seeds, while the monocotyledons have only one. The male sex organ in a flower is the stamen. Each stamen consists of a slender filament with an anther at the tip. The anther, following meiosis, produce pollen grains. The female sex organ in a flower is the pistil or the carpel. Pistil consists of an ovary enclosing one to many ovules. Within ovules are present highly reduced female gametophytes termed embryo sacs. The embryo sac formation is preceded by meiosis, hence which each of the cell of an embryo sac is haploid. Each embryo sac has a 3-cell egg apparatus, 1 egg cell and 2 synergist, 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuses to produce a diploid secondary nucleus. Pollen grain, after dispersal from the anther, are carried by wind or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tube grow through the tissue of stigma and style and reach the ovule. The pollen tube enters the embryo sac where 2 male gametes are discharged. One of the male gamete fuses with the egg cell to form a zygote, syngamy. The other male gamete fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus. PEN, primary endosperm nucleus. Because of the involvement of 2 fusions, the event is termed as double fertilization, an event unique to angiosperms. The zygote develops into an embryo with 1 or 2 cotyledons, and the PEN, primary endosperm nucleus, develops into endosperm which provides nourishment to the developing embryo. The synergists and antipodals degenerates after fertilization. During these events, the ovules develop into seeds and the ovaries develop into fruits. The life cycle of an angiosperm is shown. Plant cycle and alternation of generation. In plant, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gamete by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. This in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following pattern. First, sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plant is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, Spirogyra and some species of Chlamydomonas represent this pattern. On the other extreme, it is the type wherein the diploid sporophyte in the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to few celled haploid gametophyte. With this kind of life cycle is termed as diplontic. All seed bearing plant, that is gymnosperm and angiosperms, followed this pattern. Third, bryophytes and pteridophytes interestingly exhibit an intermediate condition, haplodiplontic. Both phases are multicellular. However, they differ in their dominant phases. A dominant independent photosynthetic thyroid or erect phase is represented by a haploid gametophyte, and it alternates within the short-lived multicellular sporophyte, totally or partially, dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. This diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alternates with multicellular sporophytic or autotrophic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most algal genera are haplontic, some of them such as Ectocarpus, Polysiphonia, kelps are haplodiplontic. Fucus and alga is diplontic.